All right, so the last part of chapter 6. In this video, we're going to be talking about the factors that change enzyme shape and how they influence enzyme activity. What happens if you change the enzyme shape? How will it affect the rate of reaction? And then how feedback inhibition is used to maintain appropriate levels of enzymes in a pathway. So just to recap, the properties of enzymes, we know that enzymes are reaction specific. So if we have one enzyme for each substrate. And the reason for that is because there's a chemical fit between the active site and the substrate. So the active site, remember, enzymes are proteins, and proteins are made up of a string of amino acids, and each of the amino acids has a different property, different chemical property. So some of them are positively charged, some are negative, some are polar, some are nonpolar. So depending on the sequence of amino acids that you have right here in the active site, that specific sequence will be able to bind through hydrogen or ionic bonding to another substrate. So if you have let's say the inside here is all like positively charged amino acids, and you have this substrate which is all negatively charged on here, that's going to act like a magnet, right? The substrate's going to come in and easily bind to the active site. But let's say if all of this in here was positively charged, and then the substrate A is a molecule that's really positively charged, as soon as that substrate A tries to get into the active site, there's going to be a repulsive force, and so the substrate won't be able to get in. So the reason why the active site fits with one substrate is because the chemical components that make up the substrate are able to bind through hydrogen or ionic bonding to the active site. Now, the thing that we need to know about enzymes is that the enzymes, right, these are the things that speed up the reaction, they're not consumed, so they can be recycled. And actually, one enzyme can actually catalyze means to speed up, thousands or more reactions per second. And the enzymes are unaffected. So they take up the substrate, they complete the chemical reaction, they help the substrate complete the chemical reaction, and then they are recycled and they can grab another substrate. But these enzymes are very affected by cellular conditions. So enzymes work at in certain temperatures, pHs, or salinity. And so as soon as you change that temperature, that pH, or that salinity, you're going to change that protein structure. And remember, the reason for that is because for our proteins, right, we know that in the primary structure, chain of amino acids, secondary structure is the hydrogen bonding between multiple kind of adjacent nearby amino acids. Tertiary structure is the binding of adjacent R groups next to each other. So positive or negative R groups are going to bind to each other. And we know that temperature, pH, and salinity can actually disrupt those bonds that hold together the secondary and tertiary structure of our proteins. And remember, enzymes are proteins. And so we'll look later at this video of how that affects our enzymes and how basically that affects their ability to function properly. Now, just a note on how to name these enzymes. Usually when you have the enzyme, it ends with ACE, so sucrase, protease, lipase, DNA polymerase. Not always, though. And they usually break down the molecule that kind of sounds like them. So sucrase breaks down sucrose. Ose is the suffix that we use when we're talking about sugar. So sucrose is a sugar. It's broken down by sucrase. Proteases break down proteins. Notice that's another type of biomolecule. Lipases break down lipids. Here's our nucleic acid. And then pepsin breaks down proteins. Remember, proteins are a chain of amino acids bonded through peptide covalent bonds, polypeptide, many peptides. So pepsin breaks down polypeptides. Okay, so some factors affecting enzyme function. First, it's enzyme concentration. More enzyme, more reaction. Substrate concentration, if you have more substrate, you're going to have a faster reaction up to a certain point. We'll talk later about temperature. We'll look at pH in this video, salinity, how much salt is in a solution around the enzyme. We'll look at activators. So basically, if another molecule here binds to this enzyme, it can activate, it can turn on the enzyme and allow it to function properly. So that's like an on switch. And then even inhibitors. An inhibitor is a molecule that binds and turns off the enzyme, stopping it from doing any chemical reactions, which can be good. Sometimes you don't want a chemical reaction to occur and you want to kind of shut down this enzyme, power off the machine for a moment. So let's first talk about substrates. So if we look here at this graph, we can see that y-axis is graphing reaction rate, x-axis is graphing substrate concentration. Substrate concentration means how much substrate is available basically around the enzyme. And you'll notice that this is the graph that we see. This is based on data that scientists have collected, looking at multiple enzymes. Notice if we have no substrate, there's no reaction, right? 0, 0. As you increase the substrate concentration, as you increase the number of substrate molecules that are around the active site, the reaction rate is going to increase up to a certain point, though. Eventually, this active site becomes saturated. Basically, nothing else can fit in. The reaction's happening as fast as it can. It can't speed up anymore. So it levels off at a certain point. So once again, as the substrate increases, the reaction rate increases. And why is that? Well, actually, the substrates here, if you have a bunch of substrates floating around, they're all moving, right? They all have some sort of kinetic thermal energy. The substrates are moving. And it actually takes the substrate needs to be moving fast enough for it to really like bump into this active site. Think of it as bumper cars. As soon as the substrate it's moving around, as soon as it bumps into the active site, then it can actually bond. But if the substrate's hanging out over here, it's not going to be able to get into the active site. So the more movement the substrates have, the more energy that they have, the more thermal energy that they have, the faster they're going to be able to bind here because they're going to be colliding. So think about the substrates and the enzymes are all moving around. And as soon as the substrate collides with the active site, boom, then it can bond and have the reaction happen. So that's why as you increase the substrate, there's more frequent collisions with the enzymes, right? So if you have, let's see, if you have like in red here, if these are enzymes, and the yellow are substrates. Think about these as like molecules, they're all moving around. And as soon as the enzyme and the substrate collide, boom, then the chemical reaction will happen. But if you only have like mostly enzymes and only a couple substrates, it's going to take a while before that substrate binds and hits and collides with that enzyme. So at some level, though, the reaction rate levels off because all the enzymes will have their active site engaged, right? So if you have a bunch of enzymes, and a bunch of substrates, eventually all the enzymes will have their active sites engaged at one time, and there's not going to be room to fit anyone else in, and so the reaction rate will level off. At that point, we call the enzyme saturated. Basically, saturation means that the active site is full, there's no ability for anyone else to get in. And this is the limiting factor of the reaction, right? Because that's stopping any more reaction from happening because all the active sites are full. So if you have a bunch of enzymes here in red, and all of them have bumped into substrates, they're all currently doing a chemical reaction inside. Even though those reactions are happening quickly, for that moment, all the spots are taken. So that's called the maximum rate of reaction. So the reaction can't happen any faster. At some level, with a certain number of enzymes and a certain number of substrates, can't fit any more substrates in the active site. Okay, now let's look at enzyme concentration. Very same thing. So if you actually increase the number of enzymes you have, the reaction rate will increase until you level off at a certain point. So the same thing, more enzymes means they're more frequently colliding with the substrates. So if you have more enzymes and more substrates and they're colliding with each other faster, then you're going to have more reaction, faster reaction. But eventually the reaction rate levels off. The substrate becomes a limiting factor. So if you have too much substrate and you have like only a few enzymes, right? You have only a few enzymes and they're all taken up by substrate, then you're not going to be able to bind any more substrates. Now, what if all the enzyme molecules can't find enough substrate? So let's say there's a bunch of enzymes, but there's not enough substrate, right? So like down here, if you have a bunch of enzymes but not a substrate, the reaction rate is slower because basically they have open spots. They have open active sites that are able to fill up with more substrates. All right, what else affects enzyme activity? Well, remember, thinking back to the fact that enzymes are proteins, and we know the different primary, secondary, tertiary, and quaternary structures of proteins, we can take a look at how, for example, temperature will affect the rate of a reaction using an enzyme. So notice here that if our temperature is increasing up to a certain point, the rate of the reaction will start to increase, right? If the temperature is low enough, the reaction rate will still be pretty slow though. However, you will reach this temperature here, which is called the optimal temperature, where the reaction rate is the fastest possible. But if you go above that temperature, even warmer, the reaction rate will slow down until you get to eventually to zero. So notice here that there is a certain window of temperature where the reactions happen the fastest. So let's think about why lower temperatures would cause a lower reaction rate. What happens to the actual shape of an enzyme or a protein if you have that protein sitting in really hot or really cold places? Notice here that the optimal temperature for enzymes in the human body is 37 degrees Celsius, and that's our temperature of our body. Okay, now let's take a look at kind of what this graph looks like in words. So like we talked about, this is the optimum temperature for enzyme activity. And why? Well, remember, we're thinking about molecules and we're thinking about temperature specifically. We're thinking about how fast our molecules moving around. So if you have an enzyme and you have a substrate, right, in order for them to actually connect and meet up and form hydrogen bonds, they're going to need to collide with each other. And so at the optimum temperature, what you're going to see is the greatest number of molecular collisions before it gets too hot. And we'll talk about what happens if it gets too hot. So for human enzymes, somewhere between 35 to 40 degrees Celsius is the best, and our human body temperature averages out around 37 degrees Celsius. So that's the reason why we have to have our body temperature at that level. And that's the reason why if you were to get sick or you were to get a fever, when your temperature increases, your body can withstand that to some extent, but if your temperature gets too high, that's going to affect your enzymes, and that could cause death, right? That's why a fever too high for too long would not be healthy for your body. So if we increase our temperature beyond the optimum temperature, what we'll see is increased agitation of molecules. What does this mean? Well, we know that protein is made up of a bunch of atoms, and if you increase the temperature, you're increasing basically their movement. They're speeding up, they're moving faster. And so within an enzyme, what that's going to do is it's actually going to disrupt bonds. We know, if we think back to our structures, we know that the secondary structure is held together by hydrogen bonds between nearby amino acids. In the same way, their tertiary structure is connected through ionic bonds or hydrogen bonds between adjacent R groups of different amino acids. And so if you heat up this molecule, the atoms are going to start moving a little bit faster. Those bonds are going to break, and basically the protein is going to fall apart, denature. So denaturation, losing its tertiary structure. So that tertiary structure, the bonds, the hydrogen bonds that are holding nearby R groups of different amino acids will fall apart. Why does this happen? Well, we know that, again, tertiary structure, our amino acids are held together by hydrogen or ionic or hydrophobic bonds, these very weak attractions. They're weak enough for if those atoms start moving up too fast, the molecule will denature. Now, what happens if you decrease the temperature below the optimum temperature? Well, the molecules are going to move too slow. Now your substrate and your enzyme, they're not going to have as many collisions. And so that's why a reaction can still occur at this rate. Notice that, right, at this temperature here, there's still some enzyme activity, but it's just happening slower because the substrates aren't bumping into the enzyme as fast. Now, notice that there are some organisms that actually can survive at really hot temperatures. So different organisms have evolved different enzymes in order to survive, depending on where their environment is. So for human, the optimal temperature for human enzymes are 37 degrees Celsius. But if you look over here, thermophiles are these bacteria or prokaryotic cells that basically love really hot temperatures. So here you'll notice 75 degrees Celsius. These bacteria, they're called thermophilic cyanobacteria. They can live in geysers. So here's a Nevada geyser, and they can live in these really hot temperatures. They're thermophiles. And the reason for that is because their enzymes have a specific structure where they're not able to denature at these really high temperatures. All right, next we'll look at pH. pH follows kind of a similar graph to the temperature graph. Notice here, this green line curve is representing one specific enzyme, pepsin. This pepsin breaks down proteins in our body that break down polypeptides. And pepsin functions at a pH of a little bit between 2 and 3. Remember, pH is a level of how acidic or how basic or how alkaline. Another word for basic is how alkaline a solution is. And so notice here that pepsin can function at low temperatures, but another molecule in our body, trypsin, functions at higher pHs. Now, these are both enzymes that are in the human body, but how is it that these enzymes function at different pHs? Well, the answer for that is that actually, depending on what part of your body you're looking at, or whatever part of your body you're in, you're going to have different pH. So your stomach pH is different than your liver pH, which is different than your blood pH, which is different than your muscle pH. So depending on where you are in your body, different pH. And so different enzymes are actually able to be used in different organs in your body. So again, as the pH changes, what happens is when we're looking at pH, we're looking at basically how many hydrogen ions, H plus ions, are in a solution. So if you have a lot of hydrogen ions, you're in a really acidic solution. And notice here that the hydrogen ions, those can easily form hydrogen bonds, or if there's a negatively charged atom or molecule in the protein structure and the enzyme structure, they can easily bind on and basically disrupt shapes. These hydrogen ions, they're like acting like magnets. They're going to go and they're going to connect with the enzyme. They're going to disrupt bonds and affect the tertiary structure. Most human enzymes exist in a pH of 6 to 8, but like we saw on the previous graph in our stomach has a much lower pH. It's much more acidic. That helps us break down our food. pH of 3, and pepsin is this enzyme that works in our stomach to break down food. Whereas trypsin is an enzyme in our small intestine, which works at a more alkaline pH of 8. All right, now let's look at salinity's effect. And remember, salinity is a measure of how much salt is concentrated in a solution. And salt is sodium chloride. Sodium chloride, when put in water, will dissociate into ions. Sodium plus, so basically a positive ion, and chlorine ions, which is a negative charge. And so again, now you have a bunch of charged particles floating around. What's that going to do to a protein? So at some level, as you increase the salt concentration, the reaction rate will increase. So actually salt can be good for a reaction up until some point. So at this point, this optimum salinity, this is like the best. And then eventually it will decrease. Why is this? Well, the protein shape, the conformation, remember conformation is a word for like basically the shape of a protein, depends on the attraction of charged amino acids. We talked about this already. As the salinity changes, what happens is it get like just like the hydrogen ions, those sodium and chlorine ions can actually add positive or negative charges to the enzyme, and now basically you've disrupted the tertiary structure and the 3D shape of the enzyme. Enzymes are very intolerant of extreme salinity. That's why we call Dead Sea the Dead Sea, because the Dead Sea has a high salt concentration. Not a lot of life can live there. It's too salty. All right, so just to recap, we've talked about enzyme concentration, substrate, temperature, pH, and salinity. You need to know these. These are the enzymes, or these are the factors that affect the rate of reaction for enzymes. Now, besides temperature and salinity and pH and some other factors, there's something called allosteric control, which basically can cause enzymes to become active or inactive. Now, when we think about this word allosteric, this word is really important. This word, if you look it up, basically means something else, something other. The root word allo means something else, allo, other, control. So this is something that's controlling the enzyme that's kind of outside of the enzyme itself. So let's look at two types of allosteric controls. The first is called an activator. The second is called an inhibitor. Okay. Now, what are these? These are molecules. They're regulatory molecules. They regulate the function of an enzyme. And what they do is they attach to something called an allosteric site. So we've already learned about helicopter. Okay, so we've already learned about the active site. Remember, the active site is the place where the substrate will bind, right, and cause the reaction to happen and turn into the reactants. The active site is different, or the allosteric site is different from the active site. It's somewhere else, allo. But what this does is it actually causes a conformational change of the enzyme. Whoa, what does that mean? Remember, enzymes can change their shape, right? If you put on a substrate to the active site, the induced fit model tells us that enzyme will kind of hug the substrate and allow that bond to form or break for the reaction to occur. But in the same way, a molecule binding to the allosteric site, this other site somewhere else on the enzyme, will cause the enzyme to shape sometime and help the reaction happen, or sometimes to slow down a reaction. So, for example, an activator will bind to an allosteric site. This is so important for you to know, allosteric site. And what this does is it keeps the enzyme in its active form, so the enzyme can actually bind to the substrate and quickly have the reaction happen. So if you look here, this is our active here. If this is the shape of our enzyme, this is the active site. So this is where the substrate can bind, and here's the allosteric site. If an activator binds to the allosteric site, then it's in its active form. But if the activator is released, then it can get into its inactive form. All right. Now, in the same way, if an inhibitor binds to part of this enzyme that can turn the enzyme off or on. So we'll look at an example on this side, basically. But the main thing you need to know is that something binding to the allosteric site will either turn on the enzyme or turn off the enzyme. Okay, so if we look here, here's an example of the active form. So the active form is stabilized. Stabilized. So basically, what this allosteric molecule does is it basically stabilizes the protein so that all of the amino acids that are within the protein are at their most stable state, their lowest energy state, in the presence of this activator. So whatever the chemistry, whatever the molecules of this activator is, when it binds to this allosteric site, it's going to cause it to be in its stabilized active form. What this means is the enzyme can work properly. However, let's say the enzyme is now in its inactive form. Well, that could be because an inhibitor has binded here to this allosteric site right here and is turned off. So now notice the active site is closed. The substrate would not be able to get in. So this is an inhibitor molecule, this is an activator molecule. Okay, now let's look again at these activators. Again, they're compounds that help enzymes turn on. Now there's a couple different types of activators. So the first type is called a cofactor. This is not a protein, it's something that's different than a protein, but it's found within proteins. And these are small inorganic compounds or ions. So, for example, if you were to have an enzyme and it were to have magnesium 2 plus, potassium, calcium, zinc, iron, or copper, these are ions. They're small atoms that are charged. These guys. These are compounds. These are ions. They're bound in an enzyme molecule. So if you look here, we'll talk more about this, but this right here is something called hemoglobin. It's a protein that's found in your red blood cells. And hemoglobin inside of it actually has iron. That's why when we're talking about your blood, we talk about iron is in your blood. That's why you can have low iron deficiency, or you can be iron deficient, which means you don't have enough iron. That's because iron's literally in your blood. And it bonds to some other pieces. It creates this thing called a heme group. Basically, it's like the thing that's connected to this iron. That's not important right now. But what these enzymes, or what these activators do, is it makes sure that the oxygen molecules can bind here correctly to the hemoglobin. Basically, it allows the hemoglobin to be turned on. All right, now another type is called a coenzyme. These are not proteins, but now they're organic molecules. So organic molecules, any molecule that's made up of carbon. Inorganic, again, these are not carbon-based, so that's why it's called inorganic. What these do is they bind temporarily to an enzyme near the active site and help turn on that enzyme. So here example is an example of a coenzyme, and it has carbon in it, so that's why it's an organic molecule. And notice here that vitamins are actually examples of coenzymes. So think about when you take vitamins, you're not taking vitamins because they're giving you energy. That's not the purpose of a vitamin. Vitamins actually help bind to enzymes and help them be in their correct shape, right, in order to help the enzyme function properly. So niacin or B3, riboflavin or B2 or coenzyme A. If you go to the grocery store and you look in the vitamin section, you're going to find these guys. They're enzymes that literally help turn, they're organic molecules that literally help turn enzymes on. Okay, now when we're talking about activators, we can also talk about something called substrate cooperativity. What does this mean? Well, if you have a substrate, it could actually act as its own activator. Okay, so this is not allosteric site, this is actually within the active site. So let's take a look here. If a substrate, so let's say this is inactive form, if a substrate bonds to the active site, it causes a conformational change in the shape of the molecule, in the shape of the enzyme, induced fit, right? We learned about this before. However, we already learned about that, right? We know that substrate binding to enzyme in the active site can change the shape of the enzyme, induced fit. But what happens is it actually helps, like let's say this is an enzyme here and it has multiple active sites. We can actually make the other active sites even more available to bind to a new substrate. It makes the enzyme even more active and effective. Let me give you an example. So our hemoglobin enzyme, our molecule over here, is made up of 4 subgroups. So if this is its quaternary structure, it has 1, 2, 3, 4 amino acid chains, polypeptide chains. And it has 4 different iron molecules here, or iron atoms here. And notice here that those iron atoms can actually bond to oxygen. So iron is literally the atom that holds on to oxygen. So one hemoglobin molecule can actually hold 4 separate oxygen gas molecules, O2. Now, if you have one oxygen that's bonded here, so one oxygen binds to the active site, actually, it makes oxygen more readily picked up by these other iron heme groups, these other iron molecules here. So the other 3 oxygens can bind even easier. So basically, one oxygen, so if one substrate binds, it conformationally changes the shape of the enzyme to allow even more oxygen to bind even more quickly. So just going back for a second, thinking about what we're learning about allosteric control. Remember, this is the control of an enzyme from another site that's not the active site, which causes conformational changes in the enzyme, allowing it to either bond better to its substrate or basically turn the enzyme off and not allow substrate to bond. Now, I want you to think of how can you remember this? How can you remember the effect of activators and inhibitors on enzymes? Well, here's a way you can remember it. If you've ever had like a friend group or maybe like a friendship, and it's just the two of you and you're really close, and maybe you meet someone one day, you go to math class or Spanish class, and someone else kind of befriends both of you, and they're just great. Like they invite you to sit at lunch with them, they invite you to hang out after school, they help you study, whatever. That's kind of like the activator, right? One of you is the enzyme, one of you is a substrate. You're helping each other out. The activator is like a third person that comes in and basically makes your friendship even better. Whereas the inhibitor is like, let's say you have a friend duo, like you and your best friend, and then there's just that third person that comes in and they're just toxic and they basically shut down your whole relationship. That's kind of like how I remember the activator and the inhibitor. As we're going through this lecture and as you learn biology, it's always a good idea to kind of create these little fun analogies in your mind to help you memorize these things. Because I know there's so many words, there's a lot going on, but the more you can kind of connect yourself to what you're learning here, the easier it becomes to memorize. Okay, so next thing we're going to talk about is we're going to look at inhibitors a little bit more closely. So again, what is an inhibitor's role? It's to regulate enzyme activity. Now, inhibitors aren't necessarily bad. Inhibitors can be good. Sometimes you don't want an enzyme turned on. Sometimes you don't want a chemical reaction to occur. Maybe you have enough of a product and you don't need to make any more product. And so if you have a lot of substrate, you have a lot of reactant, you need to shut down the reaction. You're done. But again, these are molecules that affect enzyme activity. So there is 2 ways that we can look at it: selective inhibition and activation. So we're actually going to look at we're going to break this down into 4 different things that we're going to focus on in these next few slides. So first, we're going to look at competitive inhibition, non-competitive, irreversible, and feedback. These are different ways basically of shutting down an enzyme. All right. So first, let's look at competitive inhibition. Competitive, what does it sound like? It sounds like something's competing. So let's take a look at a picture first, and I'll kind of explain. So notice here, if we have an enzyme, and this is our active site, this right here is our competitive inhibitor. It's another molecule that's basically going to take the spot of the active site. So now the substrate can't fit. It basically closes it off, shuts it off. Think of this as, let's say you're the enzyme and you have your best friend, the substrate, and you guys have always been so close since kindergarten. But you have another person that comes in and basically they befriend the enzyme. And now the enzyme has a new best friend and the substrate. They're not even paying attention to the substrate anymore. They don't care at all. Think of that as competitive inhibition. So they compete for the active site. Now, let's think of a real world example. This drug right here, disulfiram or antabuse, is actually a molecule that acts as a competitive inhibitor to help people who are overcoming alcoholism. And here's what it does. Basically, if you were to drink alcohol, you have enzymes in your bloodstream that are able to actually bind to the alcohol molecules and break them down, metabolize them, break them down so that they're not having an effect on your body. Because if the ethanol molecules from alcohol are sitting in your bloodstream, it could cause you to get really sick and really nauseous and really not great things. So the enzyme's job normally is to break down alcohol molecules and recycle them so that you're not feeling the effects of alcohol. However, if you were to take this drug, antabuse, basically what it does is it binds to the active site of that specific enzyme that's supposed to break down your alcohol, and it causes the alcohol to just sit in your bloodstream. And what that does is it causes you to become very nauseous, and it can actually cause you to vomit if the alcohol is just hanging out and it doesn't get to be broken down. And so that can help people who are overcoming alcoholism because it prevents them from wanting to drink more. All right, so what also happens is if you have this competitive inhibitor interfering with the active site, now you're going to have a higher concentration of substrate. So it's basically now there's not going to be any substrate that can bind because the solution is so concentrated with our inhibitor that basically it has taken the spots. It's outcompeted our substrate. Because we know that actually in the body there are many enzymes. So if you put a competitive inhibitor, you just need to make sure there's a lot more of it than the substrate. So that way, as these molecules are colliding, the competitive inhibitor are basically taking over the active site before the substrate can get there. So that's competitive. That's when the inhibitor takes over the active site. Non-competitive inhibitor. This is where the inhibitor binds to an allosteric site or a space that's not the active site. It's called an allosteric inhibitor somewhere else. So again, here's our active site, here's our allosteric site. And let's say this allosteric inhibitor binds to the allosteric site. What it does is it actually causes a conformation change of the enzyme. So now the active site is not open. The enzyme has changed its shape, and now the substrate can't bind anymore. Okay. So an example of this are anti-cancer drugs. You can read here. So there are enzymes that they're involved in the chemical reactions that build nucleotides. Nucleotides are the building blocks of DNA. And so if you shut down the enzyme that helps build more DNA in a cell, and you can't build more DNA, then what happens? Well, we'll get more into this later when we talk about the cell cycle. But if your cell can't build more DNA, then basically the cell can't replicate, right? Because when a cell is about to divide, it makes a copy of its DNA so that when it splits off into two new cells, now you have one cell with one copy of DNA, another cell with another copy. And this is basically how cancer works. If a cell is dividing so fast, it's replicating its DNA and then dividing really fast. Now you're going to have a ball of cells called a tumor, and that can be damaging to the human body. That's cancer. So if you can stop the DNA if you can stop nucleotides from being built, then you can stop building DNA. Then basically you can't build new cells because cells need to have DNA inside. So this can be an anti-cancer drug. Another example of non-competitive inhibitors is cyanide poisoning. So cyanide is a poison that basically binds to the enzymes that are involved in cellular respiration. So in our next couple units, we're going to talk about cellular respiration. This is the reaction that takes our food and uses enzymes to break down our food and create ATP energy for our cells. But if you were to take cyanide, what cyanide does is it bonds allosterically to the enzymes responsible for cellular respiration, turning your food into ATP. And so if these enzymes are shut down, you can't convert your food into ATP. Basically, you don't have any energy, your cells break down, your muscles break down, and you die. Cyanide. So again, we saw that if you bind allosterically, it causes a conformational shape change to the enzyme. And the active site is not working properly. It's not open. The substrate can't even bind. Okay, now the next type we're going to learn about is irreversible inhibition. So over here, these allosteric, non-competitive, and competitive inhibitors, they're not binded forever. They can actually be released and the enzyme can be restored to its normal state. But irreversible inhibition, basically, the enzyme is permanently shut down. It's called a competitor. So the competitor permanently binds to the active site. That's one type of irreversible inhibition, or allosterically. So let's say the inhibitor binds to an allosteric site and it permanently changes the shape of the enzyme. Well, now the enzyme's gone for good. And that would be really bad. So this is what happens in some kinds of like war chemicals. So nerve gas, DIPF, sarin, and even insecticides. This is how insects are killed through insecticides. Basically, they're molecules that act as inhibitors, but irreversible inhibitors. So there is an enzyme in your body called acetylcholine esterase. And this ACE, you see the ACE here, that's the enzyme. It inhibits that enzyme. So if you have DIPF, it will bond allosterically to that enzyme, and it will cause a conformation change in the enzyme. And it will not break down a specific molecule called a neurotransmitter that's necessary for your muscles to contract. So this is called a neurotransmitter. Acetylcholine is a molecule that's necessary for your muscles to contract. So when you're moving your muscles, the reason they're able to move is because your muscles are contracting. And so if you don't have the enzyme to break down this neurotransmitter, then you will not be able to contract your muscles, and basically you will be paralyzed. You won't be able to move your muscles. So that's how these gases work, by kind of basically paralyzing your body by breaking down, or not breaking down, but allosterically bonding to the enzymes that are necessary for bodily functions, like muscle contraction. Okay, the last thing we're going to look at is metabolic pathways. So sometimes when you have some reactants and you're trying to make a product, there's actually a multi-step process. So let's say you start with a reactant A, and your goal is to get to the product of G. But what might happen is you might start with A, and you use enzyme number 1 to convert A to product B, and so on and so forth. Actually, let's look at this example here. It probably is a better visual for us to see. So this is our reactant. This is our substrate, and this is our enzyme 1. This is a metabolic pathway because there's multiple steps. It's a whole path taken to basically break down, build, or break down a molecule, catabolism or anabolism. So if we have our substrate here, it's going to bind to the enzyme, and the enzyme does step 1, pathway number 1, and it's going to produce a reactant. Then that reactant is actually now going to be this product. This is now the product. This product then becomes a reactant of the next reaction with this enzyme. So notice that this enzyme is fit for this specific substrate. So this substrate will bind to this active site, conformation change, or basically the enzyme will induce fit over the substrate, causing the reaction to occur. And then here's our product, right? Our product has been created by this enzyme. Then this substrate here, this product, will now become the reactant of the next reaction, enzyme 3, right? So we're kind of following this path over here. It binds, it does a chemical reaction, it releases, enzyme 4, and then finally you have a product. Now, chemical reactions are organized this way in our cell membrane. So when we talk about cellular respiration, we're going to see this example. And what's the point? Well, actually, it creates efficiency. So now you have like an assembly line, and we'll take a look at why we might need to have that in a moment, but also control or regulation. So let's say you actually want maybe not too much product, or you want to regulate how much product is made. You can actually shut down the system earlier if you're regulating with different molecules. We'll kind of talk about more of this later when we get to cell signaling. So this is kind of just an intro, but just something to know about. There are some pathways that rely on multiple enzymes and multiple substrates to get to your final product. Okay, so let's look at efficiency. So kind of what we talked about before. We'll get more into this when we talk about cellular respiration. But if you were to go into a eukaryotic cell and you were to look at the mitochondria, remember the mitochondria is a membrane-bound organelle. So it has this phospholipid bilayer around it. And this bilayer actually has proteins in it. And these proteins look like this. So here's our membrane of our mitochondria. And there's 1, 2, 3, 4 of these channel proteins here. And these are next to these enzymes. So this right here is an enzyme. This is an enzyme. And basically, they are connected to each other. And there are multiple reactions happening at once. We're going to get so much more into this when we get into cellular respiration, but just something to kind of keep an eye on. And what they do is they link endergonic and exergonic reactions. So these chains, basically, these metabolic pathways, you're going to be able to take a molecule, break it down, and get energy, and then use that energy to build a new molecule. That new molecule can be broken down, energy is released, and now you can use that energy to build a new molecule. So we'll talk about that a lot more when we get to cellular respiration and photosynthesis. Okay, and then feedback inhibition. So we talked about these metabolic pathways, and we talked about how each product is used by the next step in the pathway. But if this last product, enough of it is made, and you actually don't need any more. Like, you're done making the packages. You have enough of this, this G, this whatever this final product is. It will actually give feedback. So feedback means it will actually go back to this enzyme, and allosterically, it'll be an allosteric inhibitor of the first enzyme, and it will basically cause a conformation change for that first enzyme, shut down that enzyme, and inhibit that enzyme. This is called feedback inhibition. Why? Well, it's because once you have enough of this product, you don't need any more. We need to stop it. We need to turn the chain off, right? Turn all machines down. So that product will now go back and stop that first enzyme by binding to the allosteric site. And what this will do is it basically allows no unnecessary accumulation of product. We've done enough. We stop production. We don't need any more. Think of it this way: if you were like in a factory and you were making something, and once you're finished, maybe there's an assembly line. Multiple people are working on this product. And at the end of the assembly line, you have like a pile, and you're piling up all the things, and you have a huge pile. Well, eventually that pile is going to get so big that maybe it goes and it knocks down the first machine that's working on it. And so the whole thing goes off because we have way too much product. We have enough. We need to shut it all down. We're done. So that's kind of how feedback inhibition works. The feedback comes from the very end. The product, there's too much product. Let's give feedback. Let's go tell that first enzyme, hey, enough is enough. Time to shut down all systems. And then if you shut down that first enzyme, well, now you can't convert A to B. So you shut the whole thing down, right? So this is called feedback inhibition. And we're going to look at this even more when we talk later in other chapters about cell signaling again. But there are some chemical reactions, like for this is an amino acid threonine. And this threonine, in order to make a new amino acid called isoleucine, actually requires a metabolic pathway where you take this original amino acid, you bind it to an enzyme in the active site, and then there are intermediate. This is a product binds to this enzyme, creates this product. This now is a reactant, binds to this enzyme, makes this product. Now it's a reactant, binds to this enzyme, makes this product. This eventually now you make isoleucine, which is a new amino acid. These are amino acids that make up proteins. Well, once this product is made, then it's going to tell that first enzyme, hey, it's going to bind to the allosteric site here, and now the active site, now it's turned off, right? Because the allosteric, think of the allosteric site as like a button. If you click it, it turns this off. It changes the shape of the active site. So now the threonine can't bind. We don't need to make any more isoleucine. We have enough of it. And that is it for this chapter. So good luck studying, and I will see you in class. Bye, everyone.